Welcome to the Prokaryotic Atlas, an expanded exploration into the fascinating world of prokaryotes. My name is Professor Triwibowo Uwono from the Department of Agricultural Microbiology at Universitas Gadjah Mada Yogyakarta, and I'll be your guide through this journey into the world of prokaryotes. Microbiology has developed through many historical stages. Antony van Leeuwenhoek was the first person who discovered living entities in the water that he named animalcules, or small animals, which was actually a protozoa. That discovery was the entry point toward the development of the more advanced microbiology, or biology of microorganisms. The discovery made by Leeuwenhoek was then followed by many landmarks of biological science, leading to the development of the more advanced system of biology, including the discovery of genetic materials in organisms. Prokaryotes are defined by their simple cellular structure. They are unicellular organisms that lack a membrane-bound nucleus and other membrane-bound organelles. This fundamental difference sets them apart from eukaryotes and is central to their evolutionary success and metabolic versatility. Despite their simplicity, prokaryotes exhibit an astonishing range of adaptations and play critical roles in every ecosystem. Organisms can be generally classified into three different entities: eukaryotes, prokaryotes, and viruses. Prokaryotes and eukaryotes are cellular organisms, meaning that their basic unit is cell, while virus is non-cellular entity. Virus is a group of entities which cannot be described as living entities, as they have strikingly different mode of reproduction. Virus is an obligate parasite that does not have an independent metabolic system, and therefore is fully dependent on the host metabolism that they invade to replicate. Viruses demonstrate their living entity features only when they are present within a suitable host cell. The prokaryotic domain is divided into two primary kingdoms, bacteria and archaea. While both are prokaryotic, they possess significant biochemical and genetic differences. A key distinction lies in their cell wall composition. Bacteria typically have cell walls containing peptidoglycan, whereas archaea lack peptidoglycan and instead have diverse cell wall materials. Furthermore, their membrane lipids differ. Bacteria and eukarya use D-glycerol and ester linkages, while archaea use L-glycerol and ether linkages, often forming unique lipid monolayers for extremophile adaptation. These differences underscore their distinct evolutionary paths. There are several ways of organizing organisms into groups based on several criteria to create a phylogeny, which gives an indication of their genetic relatedness. Due to the fast development of classification system, the phylogenetic organization of organisms may change according to the latest development. Similar to the previous slide, this picture depicts molecular relatedness between groups of organisms, which can be simplified into bacteria, archaea, and eukarya. Prokaryotes, particularly bacteria, display a variety of distinct morphologies, which are often crucial for their identification and survival. The most common shapes include coccus, which are spherical or ovoid cells, bacillus, referring to rod-shaped cells, spirillum, characterized by rigid, spiral forms, vibrio, which are comma-shaped, and spirochete, known for their flexible, corkscrew-like appearance. These shapes are not merely aesthetic, but often reflect adaptations to specific environments or modes of movement. Prokaryotes also demonstrate survival mechanisms when subjected to environmental stress. Such mechanism may be manifested by developing cytoplasmic inclusions as a storage system, or by developing endospore under dormancy condition. While most prokaryotes are microscopic, some challenge our conventional understanding of size. This slide illustrates the limits of scale, the giant bacteria. We start with Escherichia coli, a typical bacterium measuring a mere 1–2 micrometers long, completely invisible to the naked eye. Next, we have Thiomargarita namibiensis, which can be up to 750 micrometers, 0.75 millimeters, borderline visible. But the true marvel is Thiomargarita magnifica, reaching an astonishing 10,000 micrometers or 1 centimeter, making it visible without a microscope. This organism presents the anomaly. Unlike standard prokaryotes, T. magnifica packages its DNA into membrane-bound compartments, directly challenging the foundational definition of a prokaryotic cell and blurring the lines between prokaryotic and eukaryotic organization. Let's delve into the intricate prokaryotic architecture, the cellular blueprint. From the outermost layers, we have the slime coat, capsule, and mucoid sheath, which provide protection and aid in adhesion. Moving inward, the envelope consists of the outer membrane in gram-negative bacteria, the peptidoglycan layer, the cell wall, and the cell membrane, plasma membrane. Inside, the internal space comprises the cytoplasm and, in gram-negative bacteria, the periplasmic space. Key internal components include the nucleoid, containing the DNA ring, ribosomes for protein synthesis, and various enzymes facilitating metabolic reactions. The text highlights architectural efficiency. All cellular components reside freely in the cytoplasm, and the absolute lack of internal organelle compartmentalization demands extreme biochemical efficiency and rapid adaptation. This slide further emphasizes prokaryotic architecture, the efficiency of simplicity, focusing on 4 critical elements. The nucleoid is a centralized, dense region of single circular double-stranded DNA, lacking histones and a nuclear membrane. Plasmids are extrachromosomal circular DNA rings carrying advantageous genes such as antibiotic resistance, R genes. 70S ribosomes, composed of 30S small and 50S large subunits, are the production line for proteins, floating freely in the cytoplasm. Finally, the cell envelope refers to the plasma membrane surrounded by a rigid cell wall, often encased in a protective polysaccharide capsule. The overall size range of prokaryotes, 0.1 to 5.0 micrometers, is also indicated. Prokaryotes have evolved remarkable adaptations to thrive in diverse environments. This slide, Environmental Adaptation, Appendages and Motility, details these structures. Flagella are stiff, propeller-like spiral filaments of flagellin, driven by a basal body motor, enabling movement via continuous runs, counterclockwise, and reorienting tumbles, clockwise, in response to chemotaxis. Different arrangements like monotrichus, amphitrichus, lophotrichus, and paratrichus dictate their swimming patterns. Fimbriae are short, bristle-like proteins projecting by the hundreds, crucial for cellular colonization and biofilm formation. Pili are longer and less numerous than Fimbriae, including the specialized sex pilus, utilized for conjugative DNA transfer between cells. Expanding on external structures, exterior appendages, locomotion and interaction provides a closer look. The bacterial flagellum functions as a biological rotary motor, allowing directional movement through fluid environments. This complex machine, embedded in the cell envelope, uses a proton motive force to spin its filament. On the right, we see pilus, which acts as the primary channel for the cell conjugation process, enabling horizontal genetic exchange. Additionally, stalks and appendages are specialized structures that form exceptions to simple binary division. Examples include stalk-forming species like Caulobacter and Planktomyces, or appendage forms like Asticocolus and Stella, which aid in nutrient absorption or attachment. One of the most fundamental diagnostic techniques in microbiology is the principle of Gram staining, developed by Hans Christian Gram. The process involves 4 key steps. 1. Primary stain. Crystal violet floods the sample, turning all cells dark blue or purple. 2. Mordant. Iodine treatment fixes and traps the dye firmly inside the cellular matrix. 3. Decolorizing agent. Alcohol or acetone strips the outer membrane of gram-negative cells, releasing the dye, while gram-positive cells retain the stain. 4. Counterstain. Safranin targets the now transparent gram-negative cells, staining them red or pink. This differential staining is crucial for initial bacterial identification. To understand the gram stain results, we must appreciate the structural differences in the cell envelope. Gram-positive versus gram-negative. Gram-positive architecture features a thick peptidoglycan layer, 30 to 100 nanometers, anchored by teichoic acids, which structurally traps the crystal violet iodine complex. Gram-negative architecture has an extremely thin peptidoglycan layer, 4 nanometers, situated in the paraplasmic space, and an outer membrane containing lipopolysaccharide, LPS, which functions as an endotoxin. The osmotic pressure model illustrates how the rigid cell wall prevents immediate lysis or complete collapse in extreme hypotonic or hypertonic environments, maintaining cellular integrity. This slide, Biochemical Divergence, Archaeal versus Bacterial Membranes, highlights a profound evolutionary split. In bacteria and eukarya, membranes are typically composed of straight, unbranched fatty acid chains attached to a D-glycerol backbone via ester linkages, always forming a conventional lipid bilayer. In contrast, archaea possess branched isoprenoid chains attached to an L-glycerol backbone via ether linkages. This unique chemistry allows for extremophile adaptation, where opposing isoprenoid tails can fuse to form a lipid monolayer, granting extreme thermal and chemical stability, enabling archaea to thrive in harsh environments. Further illustrating the differences, architectural divergence, gram-positive versus gram-negative, provides 3D models. The gram-positive cell wall, exemplified by Bacillus subtilis, shows a thick peptidoglycan layer structurally trapping the crystal violet complex, with teichoic acids embedded. The gram-negative cell wall, exemplified by Escherichia coli, reveals an extremely thin peptidoglycan layer, an outer membrane with LPS and porin proteins. The outer membrane allows the stain to wash out easily, absorbing the red counterstain, hence the differential staining. Not all bacteria can be reliably stained by the gram method. Diagnostic exceptions, the acid-fast stain addresses this. The target group is the genus Mycobacterium. The problem is that the cell walls of these bacteria contain an unusually high concentration of waxy lipids, primarily mycolic acids, rendering conventional simple and gram stains useless. The method involves 1. Cells prior to staining, Zeil-Nielsen stain. 2. Primary stain, carbulfuxin and steam heat, drives carbulfuxin into the cells. 3. Decolorizing agent, acid alcohol, strips the outer membrane of acid-fast negative cells, releasing the dye. 4. Counterstain, methylene blue, targets the now transparent acid-fast negative cells. This distinctive staining method is critical for identifying severe human pathogens, most notably M. tuberculosis and M. leprae, which cause tuberculosis and leprosy respectively. Prokaryotes demonstrate incredible ecological versatility. Spectrums of survival. The radar chart illustrates their adaptations to diverse conditions, including oxygen requirements, aerobic versus anaerobic, temperature, extreme thermophile versus psychrophile, nutrition, autotroph versus methanogen, and salinity, extreme halophile versus non-halophile. Alongside this, they employ various reproduction strategies: simple binary division, most common, e.g. E. coli, budding, e.g. Rhodomicrobium, blastobacter, spore formation, a survival mechanism, and stalk and appendage division, e.g. Caulobacter, showcasing their diverse approaches to proliferation and survival. Prokaryotes exhibit remarkable metabolic diversity, particularly in their photosynthetic capabilities. Cyanobacteria utilize chlorophyll A and employ non-cyclic photophosphorylation, making them strictly oxygenic, oxygen-forming. Their morphology can be unicellular or filamentous chains, with standard sizes of 0.5 to 1 micrometer, but extreme variants, like Oscillatoria princeps, can reach up to 60 micrometers in diameter. They are capable of binary fission, multiple fission, or spore formation. In contrast, purple and green bacteria utilize specialized bacterial chlorophyll instead of standard plant chlorophyll and employ cyclic photophosphorylation. Their ecological impact is strictly anoxygenic, does not produce oxygen as a byproduct of photosynthesis, thriving in specialized, light-rich anaerobic environments. In summary, the prokaryotic paradigm is characterized by three key aspects. First, their structural elegance. Lacking internal organelles or a true nucleus, prokaryotes rely on immediate, cytoplasm-bound biochemical reactions. This extreme streamlining allows for unmatched replication speeds and rapid adaptation to stressors. Second, their taxonomic depth. Though historically grouped into a single monolith, modern molecular phylogenetics reveals deep, ancient evolutionary divides, remarkably positioning archaea closer to eukaryotic life than to standard bacteria. Third, their boundless ecology. From submicron spheres to macroscopic visible filaments, and from oxygen-generating phototrophs to waxy acid-fast pathogens, these organisms define the absolute physical and metabolic extremes of Earth's biosphere. That concludes our lecture on the Prokaryotic Atlas. I hope this comprehensive overview has deepened your understanding of these essential microorganisms, their unique structures, metabolic strategies, and their profound impact on life on Earth. Thank you for your attention, and I look forward to seeing you in the next class.